Okay, the fifth lecture is on fungal infections as well. So last time we spoke about Magnaportha graecii, the rice blast disease, the powdery mildew life cycle, and how fungi get into the plant. So the next section is continuing how they get in, what they do once they're in there, and the ways they destroy plant defences. So they can get in through stomata as well. Rust spores are single-celled and they stick onto the leaf by electrostatic mechanisms. The germ tubes grow over the leaf and look for stomata, and in many species they have turgor pressure. They have, stretch sensitive, turgor pressure sensitive, what they call membranes, so they can recognise curvature when their cell goes over a stomata. So obviously you've got the two guard cells, and when they're swollen and open, they are high turgor pressure, so they're going to be like that compared to the leaf surface. So they sense that, the stretch receptors in their cell membrane can sense that, and they can penetrate. So Puccinia hordae is a rust spore, and monocots have this only works in monocots obviously, but that's their host species. Monocots have predictably arranged stomata, so they grow perpendicular to the grooves, which are these lines, like this, until they encounter stomata, because if they were growing along here, they could grow and never encounter one, but if they're going like this, then it's fine. It maximises the likelihood of finding stomata. So growth in planta, there are different regions in the plant where different fungi utilise, and these are kind of determined by the risk-benefit balance. So you can have superficial growth, which is on the leaf surface. These are usually biotrophic species, so they don't massively invade, they don't trigger any response, they keep the plant alive and they feed off the live plant matter. So these are on growth surface, so there's not massive benefits. They're not taking over the plant, they're not getting major minerals, proteins and nutrients from the actual plant cytoplasm and things like that, but as it's not invasive and they use Hosteria, these specialised feeding structures, they're less likely to get detected and they're less likely to trigger apoptosis. And there's intracellular growth, which is within the cells. So if you've got cells like this, intra would be going through them and around the space between them, whereas intercellular growth is around the cell spaces. So intracellular growth releases lots of alert molecules because it's breaking up the membranes, it's causing cell lysis, so the plant is going to alert to the fact that there's chitin in it and cause local apoptosis, and it's also going to alert to its own molecules where they shouldn't be. So this is high risk but high benefit because it's releasing lots of magnesium, phosphorus, potassium, things like that from the cells. Intercellular growth is low in nutrient gain and it's low risk because the apoplastic fluid is limited in nutrients. There's some sugars, but there's very few minerals and proteins. However, the fungi can secrete cysteine-rich proteins causing cell wall leakage from space in between the cells. So there's high risk, high benefit, and low risk, medium benefit. There's also Hosteria, which is specialised feeding structures, which I'll talk about over here. So there are specialised feeding structures, as I just said, in mildew, rusts and biotrophs. They're used for nutrient acquisition. Mildews have finger-like projections, which can gain carbon, nitrogen and amino acids. This is the structure, so this is the germ tube on the outside. It creates a swelling of a hosterial mother cell, and then tiny hyphae penetrate through the cell wall into the membrane, and you have to remember that they don't enter the cytoplasm. The membrane just swells around them because the membrane's flexible. It never enters the cytoplasm, which is why biotrophs use them, because it's not killing, it's not penetrating, it's not causing any cell lysis, it's just collecting the shit. So it also has an extra hosterial matrix, and up right by the cell wall where it's penetrated, it has a thing called a collar, which is around the neck, and the collar, we're not really sure how it works, but the collar somehow alters the cell wall structure of the intermembranal structure compared to the extramembranal structure, because plants have chitin detecting molecules, and if they detect chitin, then the plant triggers apoptosis, so the pathogen, the area in which the pathogen is won't get any food, the cell will die off, sacrificing a few plant cells for the greater good of the plant, and this is the hypersensitive response. That if it senses any chitin, it just triggers apoptosis with no further questions asked. So the penetration peg, which is this bit, has different cell wall proteins. They have specialised transporter proteins, they have less chitin, they have enzymes that can break down nutrients, things like that in this bit, so it's all different throughout here. So every infection needs to spread to a new host, and this can be done by sporulation, which is sexual asexual, which is what sporulation creates the spores that are to be transmitted. As airborne over long distance, splash dispersed over short distance, runner hyphae go through the soil, and sclerotia, sclerotia is for survival, so these are the hard spores. Fungi have very complex life cycles to achieve dispersal and survival. For example, rust has 5 spore types and 2 hosts, which is beneficial to the rust because it has lots of different specialised structures to help it survive, but also easy for control because if you take away one of the hosts, it doesn't have a reserve in winter or a reserve in summer or whatever. So once the pathogen's inside the host, it can start doing things, and the 2 main mechanisms are toxin release and cell wall degradation. So many fungi have cell wall degrading enzymes which digest the cell wall, and they can remove physical obstruction or they can release nutrients. So they either break through the cell wall so they can enter, or they break through the cell wall so nutrients can come out. They have celluloses and cellulases which remove crosslinks. Through this they have endodigestion, which creates short chain, and exodigestion, which creates cellobiose. Then you have hemicellulose, which is complex in monocots, xylinases, which are pentose sugars, pectin, which are alpha-1,4 lineages. Then you have arabinases, galactinases, and manases. Proteases are degrading structural and enzymic proteins. They can help destroy the plant defense proteins like extensin. And then we also have pectinases, which degrade the pectin-containing layer of fruits, causing soft tissues. So this is Erwinia, the soft fungi has pectinases. They can get through the polyalacturonase, which breaks down sugars. We have pectin lyase, pectin methyl esterase, which removes side chains that are soluble, and they are all elicitors of plant defense response. So they all release molecules which trigger the plant to go into a response. So toxins adversely affect the host. They can kill plant cells ahead of fungal attack, which allows the fungi to get into the plant cell in the beginning. They can reduce defensibility, they can reduce competition, and they are also used by necrotrophic fungi, which are fungi that kill the plant and feed off the remains. So there are 2 types of toxicity. There's general toxins, which are phytotoxic, which is they're toxic to many species. If they infected a human, they'd kill a human. If they infected a plant, they'd kill a plant. And out of these, there are 3 main ones. So we've got tentotoxin, which causes chlorosis by disabling the chloroplast coupling factor. This releases highly reactive electrons around the cell, because if you're disabling photosynthesis response, you're just releasing the products of photosynthesis, which are really reactive, and they only become more reactive in light. So it's going to get more and more damaging until the evening when the sun comes down, basically. Oxalic acid is a simple metabolite. The hyphae are covered in toxic crystals. The crystals obviously penetrate plant cell walls and toxicity invades. Then there's serrato-ulmin, which causes wilt by stopping xylem and phloem mechanisms in the cell. Toxic crystals. Okay, and then there's host-specific toxins, which affect specific hosts and have to be present to cause disease. Without these specific toxins, the disease is not identifiable by Koch's postulates. So T toxins affect mitochondria in maize, causing sterility issues. HC toxin of C-carbonum blocks histone deacetylase. So if you're blocking histone deacetylase, you're stopping modification of chromatin and chromatin restructuring the host. So when the fungus invades, what should happen is the host can remodel its chromatin, activate transcription of some defense genes, and translate these into the necessary proteins, toxins, enzymes, blah, blah, blah. But this HC toxin blocks the function of chromatin restructuring, so the plant can't activate a defense response, and that's how it survives in the host. The AM toxin causes electrolyte leakage in some apple cultivars, and Victorin is in oats. Oats were bred to be rust resistant, but as you can see, if this is the rust locus, when they were bred to be rust resistant, they activated the other locus on the other allele on that locus, causing the oats to be hypersensitive to another rust because the two